afternoon, and I'm really very, very happy to have Dr. Aishi to give this lecture for us. I'm sorry for talking English, but you can understand, and I hope that most of you, can follow. His presentation is going to be in English, and you can make questions in Portuguese if you want, and then I translate. At the end of the presentation, I will see the questions and ask Dr. Sheikh. I'm really glad that you came and joined us because the lab of Dr. Sheikh has a long experience in virus. He has been working with many viruses that cause human disease, including dengue, West Nile, Japanese encephalitis virus. And he has been working in studying molecular mechanism of this virus replication and how this mechanism can be translated to vaccine or antiviral products. He has an impressive list of publications and citations. Not going through that. And most important, he has many projects in collaboration with pharmaceutical partners, many parts of the world. And he has recently published some key papers or collaborated with some important investigators after the SARS-CoV-2 epidemic starts. So thank you very much again for joining us, and we're looking for your conference. Thank you.
Well, thank you so much. It's such a great pleasure to join to talk about our work. I really want to thank the organizers for the opportunities to present our work. So today, I'm just going to talk about what we have done in the past year in terms of the COVID-19 SARS-CoV-2 viruses. As we all know, humans are constantly challenged by the zoonotic diseases. And these are the diseases, the pathogens used to be in the animal kingdom, and because of our interactions with the nature, and those pathogens spill over to human, and that's why we are constantly threatened by these zoonotic diseases. And then SARS-CoV-2 is just one of the examples of those zoonotic diseases. We believe it is coming from the bats. And here is a cryo-EM structure of the SARS-CoV-2. It's spherical in shape. It contains the envelope. It's double membrane indicated here in this cartoon, and it's coming from the infected cell bilipid membrane. And outside of the virion is so-called the spike protein. And the spike protein, together with its three other structural proteins, indicated here, the membrane protein, the envelope protein, and inside is the nuclear capsid, and that is wrapped around the viral genome RNA. They form the virion. And the spike protein is a trimer, a three molecule, they form a trimeric conformation. And for the spike protein, it has the N-terminal S1 and the C-terminal S2 fragment. And for the S1, it has the N-terminal domain, a receptor binding domain, which is the domain required for interacting with the receptor, indicated here, the human S2 receptor. And then that is responsible for the attachment and their enter of the infected cells. That's S1. And S2 has the fusion peptide that is responsible to fuse the viral membrane with the cellular membrane during the infection. And then there are four genera of the coronaviruses indicated here: alpha, beta, gamma, delta. And then among them, there are 7 known coronaviruses that infect humans. And 4 of them are responsible for the common cold indicated here, and then 3 of them are responsible for severe respiratory diseases, including SARS-1, MERS, and recently SARS-2. So when we started to work on the coronavirus at the beginning of the last year, actually my lab have never worked on the coronavirus before. We used to work on the flaviviruses such as dengue, West Nile, and others, and Zika. But because of the magnitude of the pandemic, and we feel obligated to work on these new viruses, which is the SARS-CoV-2. And what we started with, as I will tell you in detail in the rest of the talk, we started to focus on developing a reverse genetic system. That is the system that allows us to man-make and manipulate the viruses on a petri dish in the laboratory. And then that is very powerful. That allows us to manipulate the virus, and that allows us to study the biology, basic research, and then we can use this platform to develop tools for the diagnosis, vaccines, and antiviral drug discovery, which I will share with you for the rest of the talk. So this is how we started to work on the SARS-CoV-2. We wanted to develop an infectious clone of the cDNA of the SARS-CoV-2, because this is one of the most important tool or most powerful tool to study the virus. So we think it's very important to have it. As indicated in panel A, this is the viral genome of SARS-CoV-2. It's very long. It's about 30 kilobase long, single-stranded RNA of +sense polarity. So what we did is we were one of the 6 institutions that we received the virus from the CDC, who isolated the index case from the patient, imported patient in Seattle early last year. And we have these virus at the University of Texas Medical Branch, UTMB. And what we did is we extract the RNA from the virus, and then we use RT-PCR to amplify the whole genome, that using 7 fragments to cover the whole genome as indicated here. And then what we did is for each of those RT-PCR, we in vitro ligated them in the test tube, and then you will have a full-length cDNA that represents the copy of the whole genome of the virus. And then on the 5' end of the double-stranded RNA, we engineered a T7 polymerase, and that allows us to do in vitro transcription to make the RNA, which is identical to the viral single-stranded RNA. So once we have those RNA, if we transfect, in this case, electroporation, into susceptible cells, in this case, we electroporate them into the viral E6 cells, we're able to recover recombinant viruses indicated here by plaque assay. Those individual hosts represent viruses. And you can see this is the plaque assay from the original clinical isolates from CDC. And then this is the recombinant virus. We can, using this system, we can recover very high titer of the viruses from the in vitro transcribed RNA, 10 to the 7th plaque forming unit per mil. And this is the first reverse genetic system we developed for the virus. And then very quickly, we started to trying to use this system to develop tools for vaccine and antiviral drug discovery. What we did is we started to engineer reporter genes into the virus. In this case, what we are engineering is so-called amnion grain. It's equivalent to the GFP, green fluorescent gene, into the virus. We engineer them into the open reading frame 7 of the virus. So we can recover the virus using the same technology I introduced in the previous slide. And now we have a virus. Then when this virus infects the cell, it will turn the cell green. And this is very powerful. That allows us to do the diagnosis and the antiviral screening. On the right panel, this is the assay we use for measuring the neutralizing antibody levels in the 96-well format. So this is how the experiment performed. On day 0, we see the viral cells on the 96-well plate, and then the next day, we incubate the patient specimens with the green fluorescent virus for 1 hour, and then we lay this mixture onto the preceded viral cells, and then we incubate, let the infection go for 16 hours, and then we use the high content imaging to measure the green fluorescent cells, and then using computation, we will be able to calculate the neutralizing antibody levels afterwards. So using this platform, we tested 120 human specimens. 60 of these specimens are known convalescent serum. So those are the individuals have previously tested RT-PCR positive. So we know they are infected. We use those serum specimens for testing the neutralizing activity. And then we also use another 60 negative serum whose sample were collected before the pandemic. We know there is no a positive antibody serum positivity of those individuals. So for each of those serum, we test them against the 2 assays. One is so-called a plaque reduction neutralizing assay, which is the gold standard for neutralizing activity. And then the other is the newly developed, the reporter amnion reporter virus neutralizing assay. So for each specimen, we test them side by side against those 2 assays. You can see the numbers for each of these serums tested against these 2 assays. And then here on the right is the correlation between the conventional plaque reduction neutralizing assay and the newly developed high throughput neutralizing assay. They have a very good correlation in terms of the neutralizing antibody titers. So this is just a validation of the new fluorescent-based neutralizing assay. And then we also tested the specificity of the assay using 150 serum, which from the individuals who have been infected with different viruses or pathogens as indicated in this table, or vaccinated by different vaccines. And then these are, you can see, none of those serum cross-reacted with this reporter virus assay, indicating the specificity of the assay. So once we have validated these assays, we started to collaborate with the vaccine companies. We mainly collaborate with the Pfizer and to support their vaccine. And since these data are all published, I'm not going to go into the details. So all the preclinical phase 1, phase 2 neutralizing assay for Pfizer's vaccine, our team supported that, and we're very happy to be able to partner with them to develop the first vaccine that has been received the EUA in the US. And then also the platform has also shared with colleagues in Emory who supported the Moderna's vaccine study. Indicated here, this is the very recent publication from New England Journal of Medicine from Moderna using our amnion reporter assay to look at the neutralizing titer stability after 6 months of the second dose. You can see, the first dose here, the second dose here, and then 6 months, 3 different groups. The neutralizing titer sustained, although it dropped a little bit, but it's still substantially active against the SARS-2 viruses. And so this is the utility of the reporter assays to support the vaccine development. And also we developed another version of the reporter virus called the luciferase reporter virus. So The assay, the platform is very similar to have I already explained it before. We engineer the luciferase, in this case, a nanoluciferase, into the open reading frame 7 of the viral genome. So we can make these virus in the very high titer, 10 to the 7th platforming unit per ml. If you infect cells, in this case, viral cells, with this luciferase virus, you can see as time goes on, you have a lot of luciferase activities over the time post-infection. And these, you can see the background, it's above 6 logs over the background. And this is very robust. It can be used for antiviral drug discovery. And then using these to prove the concept, and we use this luciferase assay to test 2 inhibitors, at least in vitro, and the chloroquine and the remdesivir. You can see we infect the 2 types of cells, viral cells, A549, which is the human epithelial cells that expresses the human S2. And you can see a very nice dose response in cell culture of the chloroquine antiviral activities. And as summarized here, the viral cells, the A549 cells, the EC50, between the 2 cell lines, there is not much differences, 2 to 3-fold differences. But remdesivir, as we know, is the nucleoside inhibitor. And you can see in the different cell lines, this is the A549 and this is the viral cells, there is a significant differences in terms of the EC50s as indicated here, more than 10-fold differences between the 2 cell lines. So these results really indicate the cell types are very, very important when you're analyzing the antiviral activities. This is really because that nucleoside or nucleotide inhibitors, they require the host kinases or esterases to convert this prodrug into the active triphosphate form of the nucleoside before the viral polymerase can take it and to inhibit the RNA polymerase. So once we have validated these assays, we collaborated with Gilead to test many of the approved, the FDA approved nucleotide or antiviral drugs. And in this case, we first show you the data of the nucleoside-nucleotide analog. That's the group 1 compounds we tested. And you can see among all those nucleoside-nucleotide inhibitors, only remdesivir showed a good antiviral activities. All the others, there is no activities at all. And then we move to the second class, which is the HIV drug, as indicated here. The HIV has protease inhibitors, reverse transcriptase inhibitors, and integrase inhibitors. And among them, only nafinavir, which is indicated here, has a reasonably good antiviral activities against the SARS-2 coronaviruses. And then the third group of antiviral drugs we tested is the hepatitis C virus. And it's indicated here, protease, nucleoside, non-nucleoside, and NS5A inhibitors, and none of them have any activities against the SARS-2 viruses. And then finally, we tested the other classes of anti-infective drugs. And it's indicated here, among them, 3 of them showed some activities. Rupintravir, which is the HRV protease, serine protease inhibitors, and the antiparasites inhibitors, and anti-malaria chloroquine in cell culture showed activities. The others don't have any potential at all. So those are the work we collaborated with Gilead using the luciferase reporter-virus assay to perform antiviral drug screening. And these are all very useful, but one of the main limitation of the platform so far I talked about is that it requires biosafety level 3, right? So not many labs don't have access for the BSL-3. So the question is, can we make a system that can allow us to do authentic viral infection of SARS-2 and that can be performed at the BSL-2? So we recently published this paper indicated here that has that potential be able to perform the infection, authentic infection in the BSL-2. So here is the idea, how we did it. And we deleted 2 genes from the SARS-2 viruses, open reading frame 3 and the envelope, the 2 genes we deleted. And then we transcomplement, we made a cell line that inducibly express these 2 missing genes or proteins. So then when we electroporate this open reading frame 3 envelope deleted viral RNA into this cell line, they can transcomplement and forming the particles indicated here. And these particles are able to infect the normal viral cells. You know, in this case, it doesn't have those transcomplementing 2 missing proteins. Then they can infect, and then the RNA can start to amplify. And because these viral cells does not have the missing 2 genes, and then it no longer be able to assemble to the progeny viruses, so that it stops here. So that's basically a single round infections using these transcomplementation. And we engineered a couple of safeguards to prevent, you know, homologous recombination and other things to produce accidentally to produce the wild-type viruses. For example, we engineered the TRS from the wild-type, the transcriptional regulatory sequences, the wild-type sequences indicated here in green. We really rewired or mutated all those sequences to red, completely different sequences, so that if there is homologous recombination or recombination occurred accidentally with other viruses, these are the lethal virus. You will not produce infectious virus. It's just a safeguard engineered into our system. And then this is the slide just showing you the transcomplementing cells, which you can see this is the construct. We use the lentivirus to engineer these cell lines. We encode the M cherry just for selection of the expression. And then you can see the SARS-2 envelope protein and then the open reading frame 3. That's the 2 missing genes from the genome, deleted genome. And here are 3 clones from the cell lines we selected from the colonies from the individual cells. And you can see without doxycycline induction, there is no red cells, as this is the M cherry expression. And if you have doxycycline induction, you can see the cells turn red because of the expression of the M cherry. And then you can see the cells all turn to green, indicating they're expressing the 2 missing genes. And we also tested these cell lines are very stable. If you passage them for 20 rounds, culture these cells, and they are still very stable upon induction with the doxycycline. You can see still P20 have a very robust the M cherry expression in these cell lines. So this is the experiment, the first experiment we did. We electroporate the genomic RNA missing the 2 genes into the transcomplementing cells, and we collect the culture supernatant to look at the viral titers using the TCID50 as a measurement. You can see as time goes on, the infectious titer of the single round particles increases, but they are not great, 10 to the 4th TCID50 per mil. And under the electron microscope, we can consistently see those particles, the virion particles. And then panel B shows the single round infection. So if you use these particles to infect naive viral E6 cells for the first round, you will see those cells 24 hours, 48 hours, they tend to green, indicating those particles are infecting. But if you take the supernatant from these cells and to infect the second round of naive viral E6 cells, and you don't see any green cells coming up, indicating that supernatant from these cells don't have any infectious particle. That is supporting our single run concept as indicated in the slides. But of course, as I said, the initial titer of the system is not very robust. So we want to enhance the efficiency of this production of the neutralizing the titer of the single round viral particles. So what we did is instead of using these particles to infect naive cells, we go around to infect these transcomplementing cells. So we let these passaging to go around for 10 rounds, so that we hope that there is adaptations that is occurring through these 10 rounds of passages. And then we hope those adaptive mutations would be able to increase the efficiency of these transcomplementing system. So this is how we did. After this is the brown is the initial transcomplementing system, 24 hours, 48 hours, they're not very high. And then if you, after passaging, the green triangles for 10 rounds, you can see it significantly improves the efficiency of the titer of the single round particles. And then when we sequence these 10 round passage genome RNA, we saw 3 mutations in 3 different places, the NSP1, NSP4, and in spike gene, we have those 3 mutations. And then we started to engineer these 3 mutations into the viral RNA and to see whether we need all of them to be able to increase the efficiency of the transcomplementing system. The answer is yes. These are the black diamond symbol here. You can see it needs all 3 mutations, adaptive mutations, in order to have increased efficiency of the transcomplementing system. So then, of course, safety is a major concern in order to bring these down to the biosafety level 3. We started to test these single round particles in 2 animal models. And the first one is the hamster, which is shown here. And then what we did is we intranasally inoculate the single round delta open reading frame 3 envelope virion, as well as the Y type side by side. And then we look at the pathogenesis and the viral load of these infected inoculated animals. So here, panel B is the weight loss of those infected animals. You can see the red line is the one inoculated with 6 times 10 to the 5th, the TCID 50, the single round particles, right? And then there is no weight loss compared with the mark. And then for the Y type, even if you inoculate 10 to the 5th TCID 50, you can see significant weight loss of the infected animals. And then also panel C shows the disease over the different days after the inoculation. You can see the Y type on day 5, day 6, and day 7, they have diseases, those inoculated animals. In contrast, the single round particles, throughout, there is no disease indications of those infected animals. And when we look at the viral loading of the different specimens collected from the infected animals, the nasal wash and the oral swab and the organs, trachea and the lungs, you can see that the single round of particles using RT-PCR is multiple logs lower compared with the Y-type inoculated animals. So these data definitely demonstrate that in the hamster model, the single round of particles is highly attenuated and there is no dissemination after the inoculation into these animals. So then through collaboration with Mike Diamond's group, we further tested the safety in vivo using the mouse model, in this case, is the K18 that transgenically expressed the human S2. And we inoculated these mice using two different routes. One is the intranasal inoculation, as indicated here in panel H. After the intranasal inoculation, there is, for the Y-type infected animals, and there is a significant weight loss. And then for the single round of particles, even though the inoculation dose is significantly higher, as indicated here, there is no weight loss at all. And then survival is the same. The Y-type inoculated animal led to the death, while the high dose of single round particles, there is no death at all. And then furthermore, we went on to inoculate these animals through a different route, which is intracranial, directly injecting those particles into the brain of the transgenic mice. As indicated here, you can see the Y-type, the red one, and they have significant weight loss, and then these led to the mortality. And then the single round of particles, there is no weight loss at all. And then for the survival is the same. For the Y-type, even as low as 5 TCID50, there are certain percentage of death. In contrast, much higher dose of the single round of particles, as indicated in green triangle, and there is no death at all. So these results really demonstrate that the single round of particles in human S2 and the hamster model, there is no disease, no virulence, and no dissemination after its inoculation. So then we tested the utility of these potentially BSL-4 single round particles. And same thing, we were trying to demonstrate that this single round of particles can be used to look at the neutralizing antibody activities. Again, as indicated on the right side, we use 20 convalescent patient serum indicated here, and we test them against the conventional plaque reduction neutralizing assay, as well as the single round reporter neutralizing assays. So you can see the data are quite comparable as indicated in panel B. And then the same assay in the BS2 potentially can look at the antibody neutralizing titer. This is the monoclonal antibody, as well as antiviral drug discovery, in this case, remdesivir in human S2, and the viral cells. So these data demonstrate that the single round of particles can be used for antiviral drug discovery, neutralizing activities, and the monoclonal antibody neutralizing activity measurement. So these are just the tools for genetic tools we've developed for COVID. And now I'm going to move to the next phase of the presentation, which is the variant mutations. And as we know that initially when the spike sequence was published at the 614 amino acid of the spike protein, it is aspartic acid. It's a D614G. And very quickly, at the end of last year, the February of last year, and people report that there is a mutation. This D started to change to G. As time goes on, you can see it became dominant that these 614 become a glycine. It's a mutation. So then it really raises the question, what does this mutation contribute to? Does it increase the viral fitness? Does it increase the transmission? Or does it affect vaccine efficacy? So those are the questions we were very much interested in answering. So using our reverse genetic system, we made an isogenic pair of the viruses. So basically everything else is identical, except we mutated these D614 residue to glycine. So we can recover the plaques as indicated here. They are almost identical. And then when we perform the replication kinetics in color 3, which is the human long epithelial cells, and you can see as time goes on, the red bar, which is the mutant virus, has some advantages. And then if you measure the RNA copy numbers of these specimens from 22 to 48 hours, if the same specimen measured by RNA copy numbers, you will see actually the mutant virus RNA copy number is lower or equivalent to the wild type. So if you do the calculation of RNA copy number divided by the plaque forming unit, and this is the ratio usually used to indicate the specific infectivity of the viruses. And you can see that the red virus, which is the mutant virus, that this number is lower, indicating that the mutant virus is more infectious compared with the original viruses. So then, to further demonstrate the pathogenesis of this mutant virus, we used the hamster model to look at the pathogenesis. We intranasally infect the original D virus or the mutant G virus, and then we look at the weight loss as indicated here. You can see both the black and the red viruses, which indicates the wild type and the mutant, they all lose weight. They both lost weight to the same extent, indicating the pathogenesis, there is no differences caused by the mutation. But when we measure the viral loading in terms of plaque forming units, these are real infectious particles we're measuring. And you can see on day 2 post-inoculation that the mutant virus has some advantages, right? It's slightly higher compared with the original virus. But on day 4, you can see a significant differences, particularly in the upper airway, which is the nasal wash and the trachea. You can see it's more significant, the difference. The mutant virus is higher, while in contrast, the different lobes of lung, there is not much difference between the wild type and the mutant viruses. So these results really demonstrate that in the hamster model, that the mutant virus enhances the viral fitnesses by increasing the replication capability in the upper airway of the infected animals. And to correlate these results and to human relevance, so what we did is we started to compare the viral replication using the human primary airway tissues indicated here. So we infect the human airway culture with equal amount of the mutant and wild type viruses, and then we measure the production of the progeny viruses. You can see that the red virus, which is the mutant virus, grows significantly faster compared with the wild type viruses. And then we measure the RNA copy numbers using the same specimen, they're equivalent. And again, if you do the calculation, the RNA copies divided by plaque forming infectious viral particle ratio, you clearly see that the mutant virus is much lower, this ratio, indicating the mutant virus, again, is more infectious compared with the wild type virus. And then, to further drive down this conclusion, we perform the competition experiment. So in this case, we mix 1-to-1 ratio of the mutant and the wild type virus equal amount into the culture, and then at different days post-infection, we analyze the progeny virus population and to see what's the ratio between the mutant and the wild type virus. And you can see that this is the mutant versus the wild type RNA copy, the SNPs. And you can see that the mutant virus, if you start with 1-to-1 ratio, the mutant virus wins. And you can even start with 3-to-1 inoculant, 3-to-1, the wild type to mutant, and you can still see over the time, the mutant wins. Even 9-to-1 inoculant, as indicated in panel G, and it still can see the mutant virus wins the competition. So these results clearly demonstrate that in the human primary airway culture that the D614 increases the viral replication fitnesses in the human airway culture. It doesn't advance. Okay, so the next, what we did is we looked at the stability of the viruses. What we did is we incubate equal amount of the mutant and the wild type virus at different temperature for a certain indicated time, 42 degree, 37 degree, and 33 degree. 33 is just mimicking the up airway temperature. And then after the different incubation, you look at the infectious viruses remaining after the incubation, and you can see that the mutant virus is much more stable, thermostable compared with the original virus. We think this might also contribute to the high level of the mutant virus in the upper respiratory tract. And then after our publication of this D614G of observations and two other groups, one is from Yushi Kawakar's group and then the Rolf Barrick's group, they collaborated and they directly used the hamster model to demonstrate that the mutant virus improves the transmission in the hamster model. And then the other group that Dave Wentworth, Volker Thiel, and Martin Beer's group, they use both hamster and the ferrets to demonstrate that this mutation enhances the direct transmission in animal species. So to summarize this part of the talk, we show that the D614 enhances the viral fitnesses in the human airway, particularly in the upper respiratory tract, and that this mutation does not seem to increase the disease severity. And I didn't talk about the neutralizing activity in the interest of the time. In terms of mechanism, the structural biologist has shown that once you have this mutation, this will promote the receptor binding domain in the open conformation, like, lifted up in the upper conformation. And this is the conformation that is required for the receptor binding domain to engage the human S2 receptor to start to attach and infect the cells. So this is what we believe this mechanism is. So now I'm going to talk really quickly about our new studies on the B117 UK variant. There is a lot of studies or reports indicating that the B117, which is the UK variant, it increases the transmission and maybe disease severity, depending on which publications you read. And if you compare with the original Washington isolate, and of course we already talked about the D614G. If you compare with that, and there is 8 mutations or deletions indicated here, one by one. So what we have done is we made a panel of mutations. For example, the first one, we contain the D614 because it's all there. So this is used as the background, as the wild type. And then the first one, we engineer the deletion 6970 amino acid. The next one we deleted, there is a deletion of 145 and then 501 and the individual mutations. And then in the end, we made a combination of all these mutations into the one virus. We can recover virus as indicated here in the plaque assay. And then what we did is we used the hamster to look at the fitnesses differences in a transmission setting. So what we did is we mix 1-to-1. The wild type is the original Washington State isolates plus the D614G, and then mutant is individual mutation or the combo with all the 8 different mutations. And then we inoculate one-on-one into the hamster, into hamster one-on-one. And then what we did is on day 1 post-inoculation, we add another clean hamster into the same cage to the infected hamster. We let them co-house for 8 hours, let them contact for the transmission. And then we separate those two animals. And then we started to measure the donor animals, indicated here in panel B, and the recipient animals, which is the clean animals we put for 8 hours with the infected animals. And then we look at, in this case, I'm just showing you the nasal wash. You can see for the donor animals, after, it's a 1-to-1 ratio, equal amount, and then we look at their SNPs in this nasal wash, you can see the 501 has advantages. The mutant has the advantages over the wild type. And then the 8X, which contains all the mutations from the UK, it also has the advantages, 1.7. And then these 69 to 70 deletion, it also has advantages. In terms of recipient, you can see the same trend in the nasal wash. The 8X is 2.8 advantage fold, and then the 501 recapitulates 3.3-fold increase, and the deletion 1.5 deletion. So using this method, we nailed it down that a 501 mutation in the receptor binding domain is the key determinant for enhancement of this transmission and replication advantage. And then this is just showing you the 501 alone to do this, the competition in the transmission experiment. So this is the donor animals from day 1 to day 4. You can see starting from day 3, the mutant virus, the 501, shows advantages. In the recipient, you can see even on day 1, it has advantages. And then the others looks very similar. So overall, these results demonstrate that the 501 mutation is important for the enhanced transmission, at least in the animal, in the hamster model. And then we did the same experiment using the human airway culture. It's 1-to-1 ratio. And then we, over the time, we look at the 501 versus the wild type, as indicated here. As time goes on, the 501 mutant virus wins the competition. Mechanistically, we did the biphysics binding between the wild type receptor binding domain versus the human S2 binding. You can see the KD was over 100-fold improved once you have this 501 mutation, and it's mainly contributed by the K off. That the number is indicated here. And by modeling, we can show that by having 501 Y mutation, it increases the interaction between the receptor binding domain and the human S2. So I think this is our unpublished results on the UK B117, the mechanisms. That's how we think right now. And then last 2 minutes, I'm just going to talk about the new variants in terms of its neutralizing activities elicited by the Pfizer's vaccine. So this is in collaboration with Pfizer. We want the question is really to address whether those mutations in the spike protein recovered from the different variants, how they affect the neutralizing activities from the Pfizer's vaccine. So what we did is we made 4 viruses indicated here, and this is the original Washington state, Washington isolates. And then in B117 spike, as indicated here, it has all these mutations, and we basically swap the full spike from the Washington isolate is substituted with the UK spike. And same thing from the P1, the Brazil variant, the full spike was swapped into this Washington isolate, and the South Africa 1351 spike was swapped. And then once we make these viruses, we do plaque neutralizing assay. In this case, it's not using the reporter we just use the conventional plaque reduction assay. And these are the titers of 20 specimens from patients who have been fully vaccinated by Pfizer's vaccine. So you can see that if you use this Washington isolates, the neutralizing titer, the geometric mean is 532, and then the most concerning one, it is the South African variant, which is about a threefold reduction compared with the original vaccine sequence. So I think among those, the most concerning one is the South African variant. And then because South African variant is so important, is most concerning, and we started to look at the different regions of the South African variant. In the interest of the time, I'm not going to the details. So these are published in the New England Journal. And then last slide, which is unpublished, we just got this accepted, which is the new variants in California, B1429, and New York variant, B1526, and then the new variant, B117, plus this E484K mutation. And we did the same approach. We made those viruses, we're swapping the whole virus spike, and then you can see, in general, there is not much reduction in terms of the neutralizing activities. So in the end, I would like to thank my colleagues and the team, and these are the people in the lab, and I'm not going to go into the details who did what. And then here, great collaboration with Pfizer and the Gilead, and then some of the monoclonal antibody work with the University of Texas Health Science in Houston. And then I mentioned the Mike Diamond's team to help with the A18 mouse, and then great colleagues at UTMB, Scott Weaver, Benit Manishuri, and the funding agencies here. And I will stop here to answer any questions. Thank you for your attention.
Okay. Thank you very much, Dr. Shi. I really appreciate, and I think most of the people that are attending also like. We have time for a few questions because unfortunately we have another speaker, invite the speaker in a few minutes. But I myself have many questions, and I would like to start with I think one is important. When you use your hamster model, or you use transfected cells, do you think this can be strong enough to extrapolate to humans? Because this, I think, because you conclude many things about the effectivity, about the protective effects of the vaccines. So how is this really a good model? How it approximates, how it goes towards the human system?
No, I think that's a very good question. Nothing is able to replace human, real life, right? And I think that's why it's so important to perform the clinical studies, epidemiology studies in the clinical setting. And same thing with the vaccine. You can extrapolate the neutralizing activity reduction against the different variants, but nothing is able to replace the real world observations in human. But of course, at the same time, you need to have animal models.
Sure.
A lot of things you just cannot do in humans. So that's why complementation between the clinical study and animal models, but of course, anything you observe in the animal models, you want to see the relevance in human and validate it in patients.
I think I have so many questions, but let me try another one that I thought it's relevant to what I do. Not I'm involved in drug discovery things. When you show the data just in the beginning of your talk about remdesivir using viral cells and A549 transfectase 2, you see they're selective against one but not the other cell. How do you think that this is key for drug discovery? Because this could also be a problem. I have this problem myself because we do drug discovery, and then some components are tested against cells that are human cells but are not normal cells. What is the importance of that in the case of the viral drug discovery, in the case of the...
Yeah, I think that's a very good practical question. And in my opinion, we need to choose the most relevant, disease-relevant cell types or system to do the study. But in general, you definitely want to test, multiple cell lines or primary cultures or organoid nowadays to look at those to, validate. Because, for example, the viral cells we mentioned, the remdesivir, it's just not good. It's like a couple of micromolar EC50. But if you really look at the primary culture, like a human airway culture, it could be 1,000-fold more potent if you use those assays. And I think the Vanderbilt's team, they published this. It's very astonishing. We just have to look at what is most relevant cell types. Of course, the human airway culture, primary culture, is more relevant. And then also the viral cells, they have these transporters which will really efflux the compounds. It depends on the compounds too. So there is a lot of things will come into play, but in general, I definitely think you want to test different cell types, and then most importantly, to focus on the most relevant, disease-relevant cell types.
Okay, good. And finally, the question is regarding the in vitro experiments of variants. When you do that, you don't look for the immune response of these variants. And when the host, you have activations of cytokines, the cytokine storm in the case of the coronavirus or this virus. So, and this could affect the fitness. So, do you think this is what's the weight of that in your in vitro model? Because you do, of course, first in vitro, then you go to the hamster. But in the case of in vitro, do you expect that this will give the fitness of the virus in using the in vitro proliferation of the titers or the cell infection? Or this is a good correlation with the disease or not?
It really depends. I think that's also another good question. It really depends on the situation. And every model and the system has its weaknesses and the caveats. We just have to be careful on when we interpret the data. I think for the variants, the in vitro cell types, I think the human airway culture is much more relevant. The primary airway culture and is more relevant. And I think Carlo3 is reasonably relevant, and A549, which is a human, lung epithelial cells, which is also relevant, but those are the cell lines, right? And so we just have to be cautious on those systems. And then also, if you use the mouse model and the hamster model, and they are just not human. It's really something we need to cross-check on the different system and to make a more collective conclusions from the different systems. Okay,
so I think we are ahead of time. I'd like to thank you very much for your excellent talk. We have some questions from the students and the attendants that we hope you can send you this by mail if you like to answer. The great. Is this fine for you?
Yeah. Okay. Well, thank you so much. It's just such a great pleasure sharing our results and have this very interactive discussion.
Okay. Thank you.
Okay. Take care. Yeah. Bye-bye.
Bye.
Bye.